Everybody, this is D. Rajashekar, lecturer in Botany, Srimati Kanganeni Kalyani, Government Degree College, Vinikonda, Guntur district. In this video lecture, you are going to learn red drop, Emerson enhancement effect, and photosystems present in the plant. Let's start our video lesson. A red drop. Actually, in the last video lecture, we have come to know that plants mainly absorb visible region light, the light that is present between the lower wavelengths of light and higher wavelengths of light. Generally, this visible region lies between 360 nanometers to 700 nanometers. This visible region shows 7 different colors, shortly and popularly known as VIBGYOR, ranging from violet, indigo, blue, green, yellow, orange, and red. If you observe the rate of photosynthesis at different wavelengths and different lights, the graph would be like this, as you all see in the slide. Generally, from 360 nanometers, the plants start the increased rate of photosynthesis. At violet, the rate is a little increased. Then at indigo, it is a little more increased. At blue, the rate is taken to the peaks. Thereafter, when the plants get green light, the rate of photosynthesis is seen decreased almost completely. Why? Because we all know that the same color cannot be taken by any object. In that sense, plants, as they're green in color, they do not take green color from the outer sources. Instead, they reflect it. That is why the rate is decreased here. Then when we enter into the yellow light, again we can see little increase in the rate of photosynthesis. Then in orange, it is little more increased. Again at a red region, you can see the peaks, second peaks in the rate of photosynthesis. That is why, here in the slide, you can see the peak at a red. Here you can see two peaks. One is at a blue region and another one is at a red light region. Generally, this blue and red light are present between the 360 nanometers to 700 nanometers. Why plants absorb these two lights? Why plants perform more photosynthetic activity at these two lights? As plants are green in color, they always absorb the contrast colors to its color. They are always blue and red. That is why plants show more activity at these two lights. After red region or red light, that is after 700 nanometers, what would happen to the plants? We can see sudden fall in the photosynthetic rate after red light. This is called red drop. So up to red region, the rate is gone to peaks. Then when it crosses the 700 nanometer, that is red region, when it enters into the far red region, the rate of photosynthesis is suddenly dropped. The sudden fall, a sudden drop of photosynthesis after red light or at far red region is called red drop. Then Emerson enhancement effect, shortly known as EEE. A scientist by name Emerson conducted an experiment by isolating the chloroplastids from chlorella, an alga belongs to the class chlorophycin. Actually, when he has given high wavelength of light, that is more than 680 nanometers, the rate is little increased, that is very negligible. And when the wavelength of light is crossed at 700 nanometers, there is a drop in the photosynthetic rate, because we have already learned red drop effect. He stopped that light and he started giving another light, that is a lower wavelength of light, that is always below 680 nanometers. The rate of photosynthesis is little increased, but it is also taken as a negligible, because the rate is very little increased. Then he stopped that light also. During third time, he gave two lights together. Two lights mean a light of high wavelength and light of low wavelength together. Generally, what do we expect when something has not been, increased or done properly when a plant was given one light. How do we expect high rates in the same activity when the same plant was given two lights together? So, but to our wonder, the rate of photosynthesis has been increased double, in double amounts. Really, he also felt very astonished, the scientist who performed this experiment. When the plant was given two different lights independently, the rate was not at all increased to the expected levels, but the plant showed the increased and expected levels of photosynthetic rate when it was given two lights together. What would be the reason for this? He himself thought and came to know that the presence of two photosystems in the plant body. One photosystem has taken high wavelength of light and another photosystem has taken the low wavelength of light. As two photosystems involved in the activity by taking the respective lights, the rate of photosynthesis might have been increased. That was conclusion he has made. The so-called experiment and the so-called truth of presence of two photosystems in the plant body was named after him as Emerson enhancement effect. So, in a single sentence, we can say that Emerson enhancement effect was an experiment actually done by Emerson. By this experiment, the world has come to know the presence of two photosystems in the plant body. Now they are known as photosystem 1 and photosystem 2. Here you can see in the slide the graphical representation of Emerson enhancement effect. In the first attempt, he gave high wavelength of light, that is 700 nanometers or above 680. Then you can see the rate, how it was increased. And in the second attempt, he gave low wavelength of light, then also the rate of photosynthesis is a little increased. That is not at all considerable one. And in the third attempt, he gave two lights together, both red and infrared region. That means high wavelength of light and low wavelength of light. It can also be said like this: more than 680 nanometers and below 680 nanometers. So the rate is increased, to the expected levels. And by this Emerson enhancement effect, everybody came to know there are two photosystems existing in the plant body. They are called photosystem 1 and photosystem 2. Here in this slide, we are going to learn the major differences between these two systems. Photosystem 1 actually and photosystem 2, they both are actually chlorophyll molecules. In plants, we all know that different chlorophylls are present. But however, chlorophyll A is very important and essential and principal pigment essential for this photosynthetic activity. The rest of the pigments are accessory pigments, additional pigments, or shield pigments. We have learned it in the previous lecture. So, generally in plants, we can see two chlorophyll molecules. They are chlorophyll A and chlorophyll B. These two types of chlorophyll molecules are present more in number and also in the form of groups. These chlorophyll groups are also called quantasomes. Where do actually they present? It is given here clearly. Photosystem 1 is a group of chlorophyll molecules. Generally, they are present on the outer surface of the thylakoids. That is non-adversary region of thylakoids, free region of thylakoids. Whereas photosystem 2, opposite to that, generally this group of chlorophyll molecules is present in the appressed region, that is inner surface of the thylakoids. And photosystem 1 involves in cyclic and non-cyclic photophosphorylations, whereas photosystem 2 just involves in non-cyclic photophosphorylation only. What is photophosphorylation? Formation of ATP in the presence of light. Oxidative phosphorylation means formation of ATP in the presence of oxygen. That is seen in respiration. Photophosphorylation is seen in photosynthesis, formation of ATP in the presence of light. We know that photosynthesis is broadly studied under two heads. They are light phase and dark phase. Light phase is nothing but conversion of light energy or solar energy into chemical energy, nothing but ATP. So, the formation of ATP in the presence of light is called photophosphorylation. This photophosphorylation can also be classified into two types: cyclic photophosphorylation, non-cyclic photophosphorylation. Photosystem 1 gets involved in these two types of photophosphorylations, whereas photosystem 2 only it involves in a non-cyclic type of photophosphorylation. And another difference between them is generally photosystem 1 does not involve in the formation of oxygen, whereas the photosystem 2 involves in the liberation of oxygen. We know that in the photosynthesis equation, oxygen is liberated. We have seen it. Where does this oxygen come from? Is it from carbon dioxide or water molecule? It was proved fact that generally oxygen is come from water molecule. It was proved by Robert Hill. When water molecule is exposed to light, it is broken down into electrons, protons, and oxygen. This is called photolysis of water or Hill reaction. Generally, photosystem 1 is not related with this photolysis of water, whereas photosystem 2 is related with the photolysis of water. That is why it can give oxygen. The reaction center of photosystem 1 is P700. Generally, we have seen in the previous slide. Reaction center is nothing but chlorophyll A molecule, the principal pigment of photosynthesis. As it absorbs high wavelength of light, it is named as P700. Whereas in photosystem 2, the reaction center is always chlorophyll A, no doubt. It absorbs low wavelength of light, that is P680. And another thing is, for continuous operation, photosystem 1 must be given electrons. Those electrons come from photosystem 2 only. Whereas for photosystem 2, for continuous operation of it, the electrons should be given to it also. Those electrons come from photolysis of water. Photosystem 1 is mainly involving in the formation of NADPH, whereas the photosystem 2 is not directly involving and mainly meant for this formation of NADPH2. And another important thing is, in photosystem 1, chlorophyll A and chlorophyll B molecules are present, around or about 100. Both chlorophyll A and chlorophyll B molecules are present in 4:1 ratio. That means out of 100 chlorophyll molecules, 80 are chlorophyll A and 20 are chlorophyll B. Whereas in photosystem 2, 250 chlorophyll molecules are present. Of them, chlorophyll A and B are present in 1:1 ratio. That means both are present in equal ratio. That is 125 chlorophyll A molecules and 125 chlorophyll B molecules. In photosystem 1, carotene is present more than that of the xanthophylls, whereas in photosystem 2, xanthophylls are present more. These are some of the differences between the photosystem 1 and photosystem 2. Both are nothing but chlorophyll molecule groups. So, in this video lecture, what we have learned? We have learned red drop. Red drop is nothing but the sudden fall of photosynthetic rate after red region, that is for red region. And Emerson enhancement effect. This is an experiment actually done long back by Emerson. By his experiment, he could establish a fact of the existence of two photosystems in the plant body. Difference between photosystem 1 and photosystem 2. Photosystems are nothing but chlorophyll molecules group. They involve in light phase reactions. So that's all for this video. We'll meet in another video lecture. Thanks for your patient listening. Have a nice day. Thank you.